Hi, everyone. My name is Caralynn Collens. I'm the CEO and one of the co-founders of Dimension Bio, and excited to share with you an update of what we've been working on, focused on developing our regenerative therapeutics platform. So our goal is to restore organ function, be able to provide life-saving solutions for patients across a range of diseases. Today, we're focused primarily on liver failure. We've shown that our liver tissue therapeutic can lead to 70% survival improvement in animal models. I'll go through some of that today. And all of this is based on our BioNidum platform. BioNidum, or a nest for cells, is been validated in humans in tissue regeneration, and we've got a GMP manufacturing facility in Chicago, and now we're using that same platform to be able to deliver cells for cell replacement therapy. Our regenerative devices are partnered, out-licensed, and supported by other commercial partners. So today we really focus internally on what we're developing for tissue therapeutics. So, sorry, the formatting didn't quite translate on this. The TM is not meant to be that big, but our BioNidum platform, which is trademarked, transforms cells into these durable regenerative therapeutics. So this is a highly porous 3D printed scaffold, and it's designed to be able to seed cells into the scaffold, and then when implanted in our liver failure program, we plant this subcutaneously, so just under the skin in a tissue pocket. When it's implanted, it rapidly vascularizes. It provides for diffusion of nutrients and oxygen in the first few days, and we're able to get the density of cells that are needed for being able to replace tissue function. So as a company, we've had the opportunity to present at MESA for the last couple of years. We originally focused on our regenerative device portfolio, but then when we recognized how quickly our scaffolds vascularize and provide this nice home for cells, we really shifted the whole focus of the company towards these high-value indications. Pleased to share with you just a little bit of background on our team and some of our key advisors. Got a great group of folks in Chicago and looking to expand as we get to our next phase. So where we've been and where we're headed. So where we've been, as I mentioned, our scaffolds have been used in patients, so we have a nice safety precedent and has been through the FDA, got market clearance in a tissue regeneration product that was, the FDA actually called first of its kind. We have our GMP production facility now in Chicago, and as I mentioned earlier, our earlier assets are now partnered with commercial partners. So we really focus on where we are able to treat patients who have really lost organ function, and how do we replace or augment that function in these high-value markets. We've been really excited about the endorsement we've gotten from clinicians. As we talk to transplant hepatologists and transplant surgeons, they see a real opportunity for how we can really change the particular environment for these patients. Our initial focus is alcohol-related liver disease, and particularly a severe acute crisis called severe alcoholic hepatitis. And these patients don't have a 100% baseline liver function. Their baseline is already compromised to some extent. But what happens is they end up in an acute crisis, likely due to an alcohol binge, or there's other causes as well. And when they're in that rapid decline, what we want to do is be able to implant our therapy and rescue them from that decline. Either provide a bridge to recovery, which will be meaningful and curative in a number of different patient types, or a bridge to transplant in those that have more severe baseline liver disease. We wanted to start here for a number of reasons. First of all, this is a really big problem, one that a lot of folks don't know about. So about 150,000 hospitalizations in the US every year, and it's really a global issue. East Asia is particularly affected, Eastern Europe as well. And the 30-day mortality rates are high. There's no FDA-approved therapies. We can see 30 to 50% mortality in these patients. And unfortunately, the deaths are rising fastest in young adults, and particularly young women. So we see a real opportunity here for these patients to be able to give them something that today really transplant is the only effective therapy of being able to rescue these patients. We know as a general scientific community that liver cell replacement can save lives, but there's a couple hurdles that have prevented this from actually being feasible. One of those is that when you are talking about replacing liver function, the general school of thought is you need 10% of your liver mass to be able to supplement liver function. That's billions of cells. So we need to be able to get billions of cells into the body, keep them alive, have them function immediately upon implantation, because this is also a very quick decline in these patients as well. But then we also want to keep them alive long enough to allow for the patient's native liver to recover. And so that's really key. These patients do have some liver function, but it's compromised in this moment. And so this is the beauty of the liver. The liver has a regenerative capacity. The liver has an ability to adapt to changes in the body's environment. And so we're just really giving the liver a chance to kind of offset its function for the patient to recover. So we've done a number of animal studies now. This is one of the non-confidential versions we're sharing here, where we looked at 2 different doses of cells in our scaffold, and we're able to show in a toxin-induced animal model that we could rescue these animals from acute toxin injury. So we're looking to injure the part of the liver that is compromised in patients with severe alcoholic hepatitis or other forms of liver failure, and then be able to, again, to rescue those patients. So we've seen greater than 70% improvement in survival in these animals, which is really significant given the level of lethal injury that you can see that controls in the non-therapeutic dose, only just around 10-15% survival. So how we do this is place liver cell aggregates into our scaffold. And you can see some images here for scanning electron microscopy, where you can see these really nice liver cell aggregates adhering to the scaffold, but also giving enough room to be able to allow for tissue integration and for the vascularization that we need to be able to keep these cells viable over time. So we are in the process of scaling that density now, but we believe we're at the point where we have enough cells at the right level of density to be able to translate this to humans. All of the reasons we can do this is based on the platform that we've created, this BioNidum platform. And so a little bit of material science here. What you see here are 3 different images kind of zoomed in of our 3D printed biomaterial scaffold on the bottom. And then you zoom in a little bit more and you see the fibers, kind of the cross-hatch of fibers that are printed. Typically, when you 3D print fibers, what you're going to get are solid fibers, solid cores. And that is not what we do. We have a special formulation of biomaterials to enable these highly porous fibers. And so we get this balance of microporosity and macroporosity that looks more physiological. And that physiological porosity is what allows us to see the ability to see the right amount of the density of cells and be able to organize the cells in the right fashion, allows them to have enough porosity to overcome diffusion limitations in the first few days, allow for that early survival and resistance to an immune response, but then also allows for that space to be able to integrate with tissues over time and have that durable response due to the vascularization. So that's kind of the secret sauce behind what we do. We also manufacture our scaffolds and cells separately and then bring them together. So decoupling that manufacturing process also leads to some additional efficiencies. So just to kind of give you a sense, when you implant cells into our scaffold and then implant this in the body, we see a really close interface between the vasculature and our seated aggregates. I have some histology I'll show you as well. And we think of that as being different than some of the other methods that are being used, like hydrogels, where you don't get the right intimacy of the vasculature to be able to provide long-term durability. So we're excited about what we're able to see because we think we can give them enough of a therapeutic window for patients to be able to allow for their native recovery. The rapid blood vessel formation is really key to enabling all this to happen. And so what you see here on the left panel is our primary human hepatocytes seated within our scaffold. You see the vasculature, and then you can actually see some red blood cells if you squint, to where you can actually see the adjacency between the cells and the vasculature. And on the right, you see what we used as a control, an alginate encapsulated group of cells. Then unfortunately, after about 2 weeks, the cells began to die, and you saw these necrotic cores. And so we're wanting to prolong that survival of the therapeutic function. And so what you see in the left-hand panel here is a look at one element of liver function, albumin synthesis, protein synthesis, where we were again looking at the difference between implanting primary human hepatocytes into our scaffold versus a hydrogel control. And what we're able to see is that we can sustain that liver function over a period of time. In this case, over 8 weeks. We carried it out actually to about 16 weeks and to show that stability of function. It looked at staining for blood biomarkers for albumin, for CYP or metabolic activity, and again can see really nice robust function in the treated groups. As I mentioned in the beginning, our goal here is to actually allow the native liver to recover. And so we wanted to actually see if that was happening. And so this is in the same study that I showed earlier that showed that 70% improvement in survival. In this case, what we also did is look at the histology of the animal's liver. And so this is kind of a snapshot of animals who were sacrificed at day 4 and then at day 13, where on day 4 you can see we're getting necrosis in the right region of hepatocytes, the zone 3 hepatocytes that we would expect to see more akin to human pathology. And then in the treated group, we see that over time, we're able to, the native liver is recovering. And you can actually even see some of the inflammation that's still resident at that time, but you also see really healthy hepatocytes in that animal's liver as well. So this particular study was done with primary mouse hepatocytes within a mouse model. We've also done this with primary human hepatocytes in an immunosuppressed animal and being able to see similar function. So where we're at today, we've seen the 70% survival improvement over multiple studies now, so really consistent results. We believe that we're progressing really closely to down selection of an iPSC-derived hepatocyte to use in our system. We've gotten great feedback from clinicians as well as from payers who've helped us identify, what are those right endpoints from a clinical perspective that they're going to be looking for, and had a really great, healthy, and successful conversation with the FDA last month as well. So as we move towards the clinic, the next big focus is really scaling up to that human scale implant and being able to look at that in a large animal model, in a porcine model. Getting our product ready for clinical studies. We're looking at potential for OUS first in human studies. We've got a couple of different avenues we're looking at there, but also welcome ideas for if any of you would like to talk about potential collaborations in that regard. And really setting this up to follow the orphan drug pathways and take advantage of RMAT and other designations. The phase 1/2a trial is really going to be focused on survival as the primary endpoint. One of the reasons we chose SAH is because of this ability to look at really near-term endpoints. And so in this case, a 90-day endpoint will also look at 7-day scores as well as 30-day survival. And so I think that we've got a really quick and efficient trial to be able to move us into a pivotal after that. One of the beauties of cell therapy is, I don't probably need to tell those of you in this room, is that one product can treat multiple different disease indications. And so while we're starting with severe alcoholic hepatitis, we're also looking at where we can expand into other types of acute or acute and chronic liver failure, other opportunities to look at pediatric metabolic crises due to inborn errors of metabolism. And then beyond that, looking at how we can take our scaffold and combine it with other cell types. And so this is an area that, while we've partnered out our regenerative device portfolio, we right now have full rights to our tissue therapeutics pipeline, and so welcome partnership conversations there as well. So lastly, have to end a bit with the team and the excitement that we've got to, as we say together, do hard things to save lives. So would be pleased to talk with any of you later today or later on in the conference about what we're doing at Dimension, and really appreciate you joining us this morning. I know we're competing with keynotes, so this is good. I'm very pleased with the turnout. I appreciate that. Thanks so much.